Good afternoon, everyone. I am very pleased and honored to introduce the keynote speaker, Dr. Subra Suresh, who is the 9th president of Carnegie Mellon University, where he began his tenure on July 1st, 2013. Prior to assuming this role, he served as director of the National Science Foundation from 2010 to 2013. He is a distinguished engineer and scientist. Dr. Suresh is the first and only university president to be elected to all three national academies, the National Academy of Medicine in 2013, the National Academy of Sciences in 2012, and the National Academy of Engineering in 2002. Before joining the NSF, Dr. Suresh served as the Dean of the School of Engineering and the Vannevar Bush Professor of Engineering at the Massachusetts Institute of Technology. His research at MIT, which continues at Carnegie Mellon, into the properties of engineered and biological materials and their connections to human diseases, has been published in more than 300 research articles, 25 patent applications, and 3 books. I was amazed to read in his bio that after graduating from high school at the age of 15, Dr. Suresh received his Bachelor of Technology degree from the Indian Institute of Technology in Madras in first class with distinction, a master's degree from Iowa State University, and completed a Doctor of Science degree from MIT in just 2 years. Also described in his bio, which is posted on the wiki, Dr. Suresh's work has produced numerous highly recognized accomplishments in all the roles he has had. But I must mention one role that was not mentioned in his bio is his participation in the MSM consortium since its inception, since he is a collaborator of Dr. George Carneadakis and their colleagues, and Dr. Carneadakis' work was one of the original Multiscale modeling awards of this consortium. So Dr. Suresh is a multiscale modeler, and we are absolutely delighted that he is joining us today after literally just coming back from many weeks of traveling across multiple time zones. So he's a little tired. But we are honored that he has carved out his precious time to celebrate our 10th anniversary with us today. Thank you, Dr. Suresh. Unfortunately, he will have to leave right after his talk, so we thank you now. So now let us hear his keynote address, and please help me in welcoming Dr. Subra Suresh.
Thank you, Grace, for your very kind introduction. The work that I would like to, it's a pleasure to talk science, especially to this group, so I'm really delighted to have the opportunity, and thank you for the privilege of coming here. The work that I would like to talk about started a number of years ago, looking at the role of biological cells and the physical properties that alter the condition of the biological cells and the roles they play in the onset and progression of human diseases. So our approach and motivation to this work essentially involves the following questions. What is the role of cell properties in influencing human diseases, from a completely healthy state of the cell to a completely pathological state of a cell? The reverse is also a very interesting question, the role of diseases in influencing cell properties. As we look at biological cells, we try to focus as much as possible on human diseases. So this work started at my group at MIT a little more than a decade ago, and with a number of collaborators whose names you will see in various slides. I want to mention 2 key collaborators in the work that I'm going to talk about. Dr. Ming Dao, who's sitting here, who's been involved in this work right from the beginning, and also Dr. George Carneadakis, with whom we've collaborated for more than a decade now. In all of this multi-scale modeling and simulation, computational simulations, which are guided and validated by experiments, or experiments that are guided and validated by computational simulations, are absolutely key. And especially when we deal with human diseases, computational multi-scale modeling is vital because we can ask a lot of what-if questions in the computer that we cannot ask in the human body. That's the simple reason for it. And I will show a few examples of those kinds of questions. To put some substance and depth into it, I want to go into three major classes of human diseases. Hereditary blood disorders, and I'm going to particularly focus on sickle cell disease. An infectious disease, which is where we started our work in Plasmodium falciparum malaria, and a disease whose origins are much more complex than either a hereditary disease or an environmentally induced disease, which is looking at different types of human cancer. And we have a lot more information than what I'm able to present in the limited time that we have, but I'll try to touch on each of these things. Pretty much all of what I'm going to present here is in the published literature, so I'm not going to talk about techniques and assumptions and models, but they are in the literature. I'll just focus on the overall approach and the final results, and the details are available in the published papers. So with that, let me start with the first topic. Starting with experiments, whether we are talking about a sickle cell disease where in the deoxygenated state, the red blood cells undergo a shape change from a discocyte to a sickle shape, which causes change in mechanical properties, the mechanics of the cell, cytoadherence, and also leads to complications such as vaso-occlusive pain crisis. And in those cases, the biorheology of the cell is of importance. How do we visualize them? So I want to start with an in vitro visualization that has no chemistry or biology, but looks at the way a cell will go through a small blood vessel in microvasculature, or in the case of cerebral malaria, an 8 micrometer diameter discocyte red blood cell has to navigate through a tube in our brain, a flexible tube or a blood vessel, whose inner diameter can be as small as 2 to 3 micrometers. So how does an 8 micrometer disc go through a 3 micrometer tube while exchanging oxygen and taking back carbon dioxide and taking back to the lungs, and repeatedly doing it during the course of the 120 days that the red blood cell survives in the human body? So that's the first point. This is the work of my former PhD student, David Quinn, where he looks at a microfluidic device made of PDMS polymer, where the cross-section, which is this opening, the effective cross-section, is roughly the same as a small blood vessel in the brain. So what you're going to see in this video clip is a whole bunch of red blood cells going through the microfluidic channel, where a cell undergoes a change in shape from a discocyte to a bullet shape, and then as it exits the channel, it recaptures its original shape. And you can do a lot of very fancy modeling just with this one video, and I'll come to that in a minute. So what you're seeing here is the human red blood cells going through a microfluidic channel. There is no chemistry here. There is no biology here. There are no endothelial cells here. This is just to show the traversal of a healthy red blood cell through a microfluidic channel. So you can see here the bullet shape, and then the cell recaptures. This is a high-speed video image of this. You can slow it down significantly and use a linear viscoelastic model, and you can show that the characteristic time of relaxation for the red blood cell to recapture its shape is about 300 milliseconds or so. That can be confirmed through independent experimental tools. So this is the case of a healthy red blood cell as we visualize in vitro. Now let's go to a specific case of a human disease, sickle cell painful vaso-occlusive crisis. So in a normal hemoglobin, without the sickle cell trait or the disease, you have globular hemoglobin. But in the deoxygenated state, in people with sickle cell disease, because of the polymerization process, the red blood cell assumes a sickle shape, and that sickle shape can lead to a number of complications, which are listed here, from pain episodes to stroke to organ damage to swelling. And here is an example of a picture in a child showing the extent of swelling. And this can be done experimentally through a number of techniques, and we decided to do it in a microfluidic platform where we have precise control of oxygen concentration. So we can go from normoxia conditions with the right oxygen partial pressure with a sickle cell patient's blood to hypoxia conditions where we induce and simulate in vitro what could happen. So the first thing we show is that we can actually make the cell sickle in vitro. And by reintroducing oxygen, we can make it unsickle. And we can do it in a quantitative way so that we can take a sickle cell patient's blood and look at different densities associated with the blood and characterize based on the density fractions a pain crisis metric that can potentially be useful for drug efficacy assays, etc. So in this example, what you see on your right-hand side is a microfluidic platform where the units are given in micrometers, and then the gas exchange takes place in a controlled manner so that we can go from normoxia to hypoxia conditions. We also look at patients. The only FDA-approved drug for this case is hydroxyurea. So we take blood samples from patients who are undergoing hydroxyurea treatment and also blood samples from patients who are off HU. So we can compare the effect of drug treatment on the propensity for the red blood cell from the patient to traverse different obstacles. So essentially combining mechanics with medicine in patients' blood. So the first video I'm going to show with respect to this is, so this is blood sample from a sickle cell patient. And this is under normoxia conditions. This is what the blood sample looks like. And I would like to acknowledge our co-authors. And the middle name that you see here is Dr. Greg Cato, who's a clinician at the University of Pittsburgh Medical School. So what you're going to see in this video is that initially we start with normal oxygen conditions. And then we slowly start to reduce oxygen partial pressure. As we reduce the oxygen pressure, you will see the cells undergoing sickling. And when sickling happens, you can see the change in shape that takes place throughout. And then we start to reintroduce oxygen, and you will see the time for unsickling. So that's what this video shows. So we start with normoxia, and now we start to take, and you can already see discocyte cells sickling to form sickle shapes. And they also, because of the polymerization process, they also induce tethers that are sized for increased cytoadherence. And then at this point, roughly, we start to reintroduce the oxygen. And then very quickly, we go back to the initial shape. So we can do a lot of quantitative things. For example, the time for sickling is almost 10 times the time for unsickling. And that has direct correlation with a lot of clinical outcomes. So we can try to do that in an in vitro assay using these kinds of tools. Now, how does this change in shape and the hypoxia condition affect a patient? So that can be demonstrated in a very simple way in vitro in the next video. So what you see here is a series of obstacle courses that we put in for the red blood cell. So from your right to your left, the red blood cells are being pushed through under controlled partial pressure conditions. So as they go through the obstacle course, a healthy red blood cell or a red blood cell from a sickle cell patient under normoxia condition will be able to go through. And then a little bit into the video, we're going to reduce the oxygen partial pressure. And what you will see on the right-hand side at the entry point is that the cells will lose their ability to go through this obstacle course. They'll get stuck. And this is sort of an in vitro demonstration of the way in which pain crisis will begin to manifest itself in the human body. And there are lots of details that I won't go through, the effect of hydroxyurea, the effect of hemoglobin concentration, etc. So right now we have normoxia conditions. and you see the cells going through. And then now we start to take the oxygen, reduce the oxygen pressure or oxygen concentration. So under these hypoxia conditions, you can see the cells begin to get stuck at the obstacles. And you'll see as time goes on a little bit, there is an incubation time involved in this, and you can see more and more of these get stuck. The moment we reintroduce oxygen, they'll release themselves very quickly and regain their ability to navigate through this obstacle course. So this sort of gives you an idea of the capacity of the tools that we have. So now we can look at sickle cell blood from a particular patient and characterize them into different density characterization using centrifugation methods. And using that to quantify delay time for cell sickling and also sickle fraction as a function of density fraction under with and without hydroxyurea treatment. And you can get some quantitative trends out of this in a very systematic way. So these are all experimental things that give us opportunity to develop very detailed quantitative tools and models using a variety of approaches that are available to us. So as you've heard from George Kaniadakis, he and his group, we've been working with them for almost a decade now, looking at dissipative particle dynamics as one tool to try to address to quantify and add to the experimental part, but also guide the experiments and do things computationally that we cannot do through experiments alone. In the DPD particle simulation, of course, we can take clusters of atoms, molecules, and the particles interact through pairwise potential. And this has been calibrated. We've done so many experiments using so many different techniques for red blood cells, and others have done that in the literature. So we can calibrate it for a variety of purposes. I don't want to go into the details of this because that's a topic that George has talked to a number of you about previously. But instead, I want to focus on some of the recent results. The experiments that I just showed you for sickle cell disease, this is the work of Lee, who works in George's group. The same experiment that I showed you, this is the computational simulation of that experiment so that we can try to calibrate the model, but then use the model to do predictive things. So let me show you the computational version of the real experiment that I showed you earlier. So this is what we get out of this simulation, and we can induce shape changes. We can induce a variety of other things. We can induce cytoadherence, if you will, if you have reliable experimental data to validate the model. The second example that I want to show is the flexibility of this model. So you can, using the experiments to guide the model, you create the appropriate sickle shape, and that can be done through a variety of means, including stretching the polymers to a certain level within the sickled red blood cell. And then you can do the biorheology in the simulation. So here is an example of the capability of the DPD model to facilitate for a single cell as it goes through a channel. In this case, a fairly large channel compared to the diameter of the cell. Now let me take it one step further. How do we go from mechanics of the cell to mechanics of an organ? The human spleen is the largest lymphatic organ in the body, and the study of the mechanics of the spleen is quite tricky, as you know. So this is an example of a situation where multiscale modeling and computational tools are enormously helpful to guide not only in vitro experiments or in the reverse direction, but equally a number of ex vivo experiments using the real human spleen. I'm going to come to that in the next slide. The human spleen is a very complicated geometry involving the red and the white pulp. And the real fitness test for a red blood cell in the human body, the most stringent fitness test, is the interendothelial slit in the human spleen. How the red blood cell navigates through the interendothelial slit determines a lot of things. So this is a work that was published last year through collaboration with George and also Pierre Buffet, who's a clinician scientist at Institut Pasteur in Paris, with whom we have had the pleasure of collaborating for a number of years. So the work involves four different approaches. The first approach is to take an axisymmetric model, a simple idealized axisymmetric model, and we take a red blood cell and force it to squeeze through the real geometry of an endothelial slit, whose geometry has been characterized using high-resolution microscopy by a number of groups, most notably the group in Institut Pasteur in Paris, which is what you see on the lower right side. The second experiment is to see if we can do DPD simulations of a healthy red blood cell going through the interendothelial slit, so we can make the predictions on the cell area and volume that are required to make the cell traverse through the interendothelial slit. The third experiment we did is a real experiment where some patients who have benign pancreatic tumor, in order for the surgeon to reach the pancreas, in some cases, they need to take the spleen out, even though the spleen is perfectly healthy. So when the spleen is removed from the human patient, the ex vivo spleen is fully functioning. Now, because it's outside the body, you can do a lot of things with it. So one of the things that Pierre Buffet and his colleagues have done is the isolated perfused spleen experiment, where you can send the malaria parasites through the spleen. You can do the analysis of what the parasite does to the human spleen, how the spleen filters red blood cells and the parasites, and what is the mechanics of the spleen itself. And the fourth experiment they've done is they've also taken a synthetic spleen developed through a combination of beads, and the synthetic spleen was used to simulate the real human spleen. Now we can do DPD simulations of the real spleen and the synthetic spleen, and we guide their experiments. Out of this comes a whole wealth of experiment, which is what we tried to publish last year in this paper. So here are putting it all together. On your lower left is the real human spleen that's taken outside the body, where they do the isolated perfused human spleen system, where we can do sickle cell tests. We can do there's been a lot of work done with Plasmodium falciparum malaria. Your top left is a synthetic spleen developed at Institut Pasteur, where they've calibrated and perfected the design of the synthetic spleen to match the real human spleen, so they can do in the synthetic spleen experiments that cannot be done in the real spleen. To the right is our simulation guiding both the synthetic spleen and the real spleen. At the top right is the DPD simulations of how a healthy human red blood cell will go through a synthetic spleen that's designed by Institut Pasteur in Paris. In your lower right is the DPD simulation of how a healthy human red blood cell will go through the interendothelial slit in the real human spleen, but it's a computer simulation of that. By putting all of this together, we can refine the design of the synthetic spleen and ask a lot of what-if questions on how to understand the mechanics of the human spleen itself. So let me show you this figure that comes out of this. So in the late 1960s, there was an excellent paper published by Canham and Burton, who took a large number of healthy human red blood cells from human subjects and tried to characterize their area and volume. So the surface area of a red blood cell and the volume of a red blood cell, and they got a whole bunch of data. And they postulated, based on a very simple analytical model, that in order for a cell to be healthy, the surface area versus the volume of the healthy red blood cell has to be determined by the narrowest fitness test in the human body. And they argued that the narrowest fitness test in the human body for the red blood cells is the smallest blood vessel in the human body. So they did an analysis, and they got this data, and they had the conclusion that it was the smallest blood vessel that determines the surface to volume ratio of a healthy red blood cell. If it is off of that, for example, if a patient has spherocytosis, the surface to volume relationship will be different. So that's what they found. But when we did our experiments using their data and more recent data by Gifford et al., and then we did our analytical model using axisymmetric model, the dashed line is the result of our analytical prediction, and the purple line is the predictive result of our DPD simulations. We found that the most stringent fitness test in the human body is not the smallest blood vessel, it's the interendothelial slit. So if the blood cell cannot meet the surface to volume ratio requirement of the interendothelial slit, it won't be able to navigate through the spleen. So this is based on a lot of experimental work and characterization work and DPD and a very simple analytical model. Let me just say a few things before let me just show two other examples of this combination. In the case of an infectious disease, if you look at Plasmodium falciparum malaria that happens in the bloodstream of the humans, when a malaria parasite or a merozoite, Plasmodium falciparum merozoite, invades a healthy human red blood cell in the bloodstream, it gets inside the red blood cell, and within 48 hours, the cell undergoes a number of changes, property changes. The most visible changes are the significant stiffening of the cell during the course of the 48 hours, especially in the first 24 hours, called the ring stage. And then in the second 24 hours, there are nanoscale knobs that form on the surface of the red blood cell that cause significantly increased cytoadherence, which causes the cell to stick to microvasculature. Then after about nearly 48 hours, the cell ruptures and spills the parasites, which are multiplied, into the bloodstream. And that's when people with malaria usually see a spike in that body temperature as the immune system tries to fight the parasite. So the two critical factors are increased stiffness and increased cytoadherence, which are both mechanical in nature. And that's why mechanics plays a very important role. So about 10 years ago, my PhD student, John Mills, and a number of collaborators and we developed an experimental tool to measure the stiffness, and we showed at that time that the increase in stiffness of an invaded red blood cell invaded by Plasmodium falciparum, is anywhere from 10 to 100 times that of the healthy red blood cell, using laser tweezers. And this has then we can do a lot of things. So when the Human Genome Project came into existence, we were able to identify a particular protein called RISA, ring-infected erythrocyte surface antigen. We could knock out that protein from the parasite, and then when we repeated the experiment, comparing the invaded red blood cell without the RISA protein with the wild type, we were able to show that one protein was responsible for about 3 to 4 times the increase in stiffness. And we were able to reproduce it by knocking it back in. So what you see here in the top row is a healthy cell that can stretch, and this is a very tricky experiment to do, and you need 150 piconewton force. That's why you need laser tweezers in a microscale system in a fluid medium. In the lower row, you see an infected cell which completely loses its ability to stretch even under a high force of 150 piconewtons. So now you can demonstrate that this change in stiffness will be one of the contributing factors for things like cerebral malaria. So here is our hypothesis. If you have a small entryway to the two most fatal forms of malaria in humans are placental malaria in pregnant women and cerebral malaria, especially in children. So here is the hypothesis. If we were able to visualize in the human brain, which we cannot at the cell level at this point, this is an in vitro demonstration of this. If you have a number of red blood cells that are invaded by the parasite and stiffened by the parasite, at the entryway to a small blood vessel, and this is an effective 2 micrometer diameter blood vessel. So this cell is infected with a malaria parasite, which you see here, and this cell is infected with a malaria parasite here. Our hypothesis is that these two cells will not be able to go through the small blood vessel, whereas the other cells around them will be able to. So in this video, we demonstrate that. So this is in collaboration with my colleague, C.T. Lim from the National University of Singapore. And now you can see those two cells cannot go through the blood vessel, whereas the others around them. If you had 20 such cells blocking the entryway to a blood vessel, the surrounding tissue may not get oxygen. So that's the point of this demonstration. Let me just close in the last 2 minutes or so with a very recent study looking at role of multiscale modeling in detecting rare circulating tumor cells. This is some work in collaboration with Dr. Tony Wong, who's now at Duke University in the Department of Biomedical Engineering. So circulating tumor cells, of course, are very difficult to track and capture. And most of the current techniques, there are more than 300 clinical trials currently in the US. The current techniques that are available for them are centrifugation, FACS, attaching magnetic beads, and different types of chemical and biological labeling, filtration, dielectrophoresis, etc. Each of these techniques leads to a downside, which is when you capture the cell, there is an artifact of the experiment that affects the properties of the cell. So the question is, can you capture the cell with as minimal an artifact as possible? So Tony and his group, and in collaboration with us, we developed this technique together for using surface acoustic waves. The idea behind this is we know that ultrasound is very benign on humans. Hundreds of millions of women over the decades have used ultrasound during pregnancy. Can we use sound waves at the cell level now to modulate and separate that rare circulating tumor cell so that we can capture the cell, hopefully without any artifact? I won't go into the details of the technique other than to show that if two cells, two particles of the same material, but slightly different sizes, go through a microfluidic medium between two IDTs, or interdigitated transducers, that produce the sound waves, the difference in pressure will cause the larger particle to go one way and the smaller particle to go another way, so we can separate them. If it's two particles of the same size but different material properties, different properties can be used to separate them. So that's the idea. If that is the case, you have a cancer cell and a healthy white blood cell, we can still separate them because of either change in size or change in properties. So we did a lot of studies combining acoustic properties and physical properties, and we were able to separate them. I won't go into the details of this other than to show you the result. The modeling guides the design of the experiment. So here we have cultured cells. Cancer cells go in one direction and healthy cells go in a different direction, and we can optimize it through multiscale modeling. Then we went to Hershey Hospital in Pennsylvania and got blood samples from 3 patients who had breast cancer, and also used a lot of cultured cell experiments, and we could detect 1 in 100,000 cells, that's a cancer cell, out of 100,000 cells, with an accuracy of about 84%, and we are trying to improve that accuracy. And we've shown that these captured cells have minimal artifact because we are using low energy sound waves. So with that, let me conclude. I've shown 3 examples of sickle cell disease, a hereditary disease, malaria, and in the case of a circulating tumor cell, and understanding human diseases through the benefit of quantitative experiments, computational simulations, and especially connections to clinical relevance is very important. That's why in each of these projects we have at least one clinician as a co-author in our papers. The potential benefits of this are increasing our fundamental understanding, mechanistic understanding, improved diagnostic tools, and hopefully novel therapeutics. And let me leave you with this slide, and these are various colleagues and collaborators in different institutions who have significantly contributed to this work. Thank you.